to our Unit 4. Oh, shoot, sorry. Welcome to our Unit 4 CT project. I'm Lily.
I'm Crystal.
I'm Madison.
And I'm Dua Sheikh. Our enhanced sense is super smell for cortisol and stress-related hormones. Our improved sense is an enhanced form of human chemoreception inspired by service animals. It allows humans to detect stress through scent. This allows early detection of panic, medical, or emotional distress before visible symptoms occur. The olfactory epithelium, olfactory bulb, and orbitofrontal cortex work together to detect and interpret stress and cortisol-related signals. The epithelium senses these chemical cues. The bulb organizes the information, and the orbitofrontal cortex interprets it, allowing someone to quickly recognize when another person is stressed. The olfactory bulb is enlarged to include special glomeruli that detect cortisol and stress-related chemicals. This expanded structure increases the speed and accuracy of processing these signals and strengthens the connection between the nose and the brain, allowing stress information to be transmitted and interpreted more efficiently.
So for integration and homeostasis, our enhanced super smell uses modified olfactory chemoreceptors that detect airborne cortisol and stress-related hormones. So sensory signals travel through the olfactory nerve to the olfactory bulb, to the limbic system, and then to the hypothalamus. This pathway allows the new sense to integrate with the autonomic nervous system by adjusting alertness and emotional responses. The somatic nervous system responds by guiding voluntary behaviors such as choosing the approach or distancing yourself from someone whose stress hormones you detect. So, a possible trade off could be increased energy use and cognitive load as the brain constantly is analyzing emotional hormone levels, or mild sympathetic activation may occur, raising alertness but risking emotional fatigue.
Okay, so next we have the mechanistic and scientific explanation. First, we have stimulus detection or chemoreception. So, in our design, the stimulus is stress related hormones, specifically cortisol, which is released through sweat and skin secretions. These chemical molecules enter the nasal cavity and then bind to specialized chemoreceptors in the olfactory epithelium. And then these chemoreceptors are designed to detect small changes in chemical concentration, which makes them biologically ideal for detecting hormone fluctuations. And then we lead into transduction and receptor potential. So once cortisol molecules bind to receptors on the olfactory sensory neurons, chemical transduction occurs. This causes chemically gated ion channels to open, allowing sodium ions to enter the neuron. This creates a receptor potential, which is a graded potential whose strength depends on how much cortisol is present. And then a higher cortisol level creates a larger receptor potential. The third process then leads to action potential generation. So if the receptor potential reaches a threshold at the axon hillock, a voltage gated action potential is generated. This follows the all or none principle and rapidly travels along the sensory neuron toward the brain. In our design, increased myelination allows saltatory conduction and increases detection speed. And then next, we have the afferent sensory pathway to the brain, which is the action potential travels along an afferent sensory neuron and enters the CNS and then is relayed through the olfactory nerve, so cranial nerve 1. And then the signals move directly to the olfactory bulb, then to higher brain centers, and then this makes hormone detection fast and emotionally responsive, which is important for emergency stress detection. The fifth one, integration in the brain. So after synapsing in the olfactory bulb, signals are sent to the limbic system and hypothalamus, which are responsible for emotion, stress regulation, homeostasis. The cerebrum then integrates the sensory data, and then it can interpret the cortisol concentrations as patterns or signs of danger, panic, PTSD response, or medical distress. And then the final one that we have is projection pathways and behavioral responses. So projection pathways send the process signal to motor and autonomic centers, triggering an appropriate response such as alerting people nearby, physically guiding the distressed individual away, activating emergency systems, or initiating calming physical contact. This mimics how service animals instinctively respond to human stress cues, but through enhanced human sensory projection pathways instead.
Okay, and then for the creative and realistic application, so we based this off of the medical dogs and service dogs who can have that sense of smell for detecting when their owner needs help and assistance. Like a lot of people with epilepsy may have service dogs to determine when they'll have a stroke before they have it. That could act as a similar concept where um, the sense of smell would be able to detect when somebody's body is having hormone changes that will lead to a medical attack, like a heart attack or a stroke or seizure. Seizure, that was the epilepsy one, not a stroke. But um, yeah, so this would be very helpful in the medical field because it would contribute to helping not only prevent a heart attack, stroke, seizure, whichever one it may be from happening, but it also could have um quick response time to the medical issue. Sometimes when it already happens, a heart attack or a stroke or seizure, it's too late to recover from that. But this would allow for there to be signs before time, noticing the hormone changes with the sense of smell like surface dogs do, and that would be able to help people survive more heart attacks, seizures, strokes, etc. And all around, just if someone's in emotional distress, it would also be helpful in detecting that and help calm them down before they get overwhelmed and cause a panic attack or anything like that. Thank you. That's it for our presentation. Any questions?
Yes. How would this sense differentiate between threatening stress and normal life-related stress? And who would this super sense be given to?
So for the differentiation, the hormones act similarly because the body can't differentiate between like life or death and normal, but how long it lasts and how much of the hormone is going to determine like the life-threatening and the difference between the life-threatening and the non-life-threatening, just the emotional stress. So if you can smell like more hormone for a longer amount of time, that means it's more life-threatening than it would be if it was just normal stress. And then the super sense would probably be given to medical professionals who are around people who are more prone to like having those instances where it would be harder for them to recover afterwards. And you would want to react a faster reaction time before.
What happens to the receptor potential when there's an extremely small amount of cortisol detected, and how does the axon ensure that the weak signal is still transmitted, the information reliably?
So, when only a very small amount of cortisol is detected, the receptor generates a small receptor potential. And since receptor potentials are graded and they reflect stimulus strength. On its own, the weak signal may be subthreshold and won't automatically trigger an action potential, but the axon ensures reliable transmission through temporal and spatial summation, where multiple small graded potentials add together at the axon hillock. So once the combined signal reaches threshold, a full all or none action potential is generated. And because action potentials do not decrease in strength as they travel, the signal is transmitted reliably and accurately to the brain, even when the original stimulus is extremely weak.
So this is more like a general one. So obviously there's smells in the air, like sweet smells, savory smells. So how does the smell of cortisol get from like our human body to the air?
So, when someone is stressed, their body releases cortisol linked like chemicals and hormones through like their sweat or their like oils on their skin. So the chemicals can be like pretty strong, so they can be detected in the air. And in our project, the olfactory system is modified to sense when these stress-related chemicals occurs. So it doesn't sense the cortisol directly, just the cues and the stress-related like chemical cues.
I just had a question out of curiosity. I'm just curious how the body would maintain its internal cortisol levels. So like, would it treat the external cortisol levels as its own, or is there a sort of mechanism to prevent that?
So no, the body wouldn't treat external cortisol as its own stress because our sense is detecting cortisol in the air, but the cortisol never actually enters our bloodstream. And because of that, the hypothalamus can tell the difference between external cortisol signals and internal cortical signals. So internal cortisol is still controlled by the hypothalamic pituitary adrenal axis through negative feedback. So your body doesn't accidentally raise your own cortisol just because you smelled someone else's strength, smell somebody else's stress because of this.
Yeah, that's it for our presentation, and I'll switch screens so you guys. Whenever you guys are ready.
Okay. Hi, my name is Aaron, and today we'll be presenting our Unit 4 CT project around UV vision. Our project is also with Daniela, Guadalupe, and Andrew.
Okay, so for UV vision to work, we need to modify four main structures: the cornea, the lens, the retina, and the visual cortex pathways. Normally, the cornea and lens block UV light to protect the eye, so we would replace them with an artificial lens that will safely let UV light through. The artificial lens would be placed surgically. The new lens system would also include muscles that could block UV light when it is needed. Next, we will add a UV-sensitive cone to the retina using gene and cell therapy. Humans usually only have 3 cones types that are red, green, and blue. But with a UV cone, that will allow the retina to detect ultraviolet wavelengths and send those signals through the usual bipolar and ganglion cells into the optic nerve. Finally, the visual cortex, when it is B1 and B4, would develop a new UV color channel so the brain can interpret the new type of information. Thanks to neuroplasticity, the brain will gradually learn how to process and recognize UV light. So overall, UV vision requires multiple changes to both the eye structure and the brain visual pathways. Now, these images basically show how the process would work with the different like lens and the parts of the eye that will be modified.
Okay, so the control of the system is purely involuntary, being controlled by the autonomic nervous system. So these new supravior muscles that would be innervated by the oculomotor nerve, um, which would relax and that would keep the UV blocking lens behind. And then when a threshold is reached, then they'll constrict and lift up the lens. So if we go to the next slide, I'll explain that, and then we can go back. So right there, like you kind of have like a system on top that can retract this UV blocking lens up when you want to see UV light, and then when it's too much UV light, then we drop back down. And then we can go back to the last slide, please. And then so because of this, the trade-off would be decreased quality of vision, like normal vision, because even though like there's a threshold to drop the UV blocking lens to protect the rest of the eye, when that lens is open and UV light is getting through, like there's still going to be damage occurring. So um, there'd be a thin layer of crystalline proteins on like the normal rods and cones to protect them from the UV light. But yeah, this would decrease the overall quality of vision.
So when we add the UV vision to our eyes, the body's going to have a different homeostatic process to help with this. So one of them could be increased melanin in the eye structure, and it's going to need this because the melanin is going to help protect the eye from getting damaged from UV light. So we're going to have so the inner structure in the eye could be like the cornea or the retina can have more melanin, especially the iris and the choroid. And then there's going to be an enhanced melanocyte activity to increase this melanin. And then another process we have is the energy usage. So due to this new type of the visual cell being implemented, we're going to need the new muscle group and increased melanocyte activity. And then there's going to be an increased amount of energy usage because of this. And then on the big pictures, it may not be a huge difference, but it does affect the homeostasis a lot.
And our creative application that we applied our UV vision to would that it would be helpful in the medicine and medical use because UV light is often used to sterilize, and UV vision would make it easier for someone to see areas that have not been properly sterilized, seeing germs in like hospitals and clean laboratories for just a cleaner environment. Another is safety and search, and being able to see the fluorescence would make it easier for someone to see blood or fingerprints, and it would kind of be almost like a forensic help tool. Thank you. Does anybody have any questions regarding our slides?
Yeah, so my question is, how would the brain adapt to interpreting a brand new type of cone that humans don't naturally have?
Okay, so to answer your question, the brain would take time to adapt to interpreting a new UV cone, but it is possible because of the process called neuroplasticity. Neuroplasticity allows the brain to adapt, learn, and recognize itself and form new connections. And in this case, the connection would be to process UV information. So as long as the UV cone is sending signals through the normal visual pathway, the brain will gradually learn to interpret those signals and develop a new color channel. So over time, it will build a new perception of UV light, allowing UV vision to occur.
I have a question. How would the body maintain homeostasis when new UV cones are added?
Yeah, so kind of like the other homeostatic process, the body's just going to naturally adapt to these new UV cones. One way that we didn't really talk about is the neurointegration along with these UV cones with the homeostatic process. So adding a fourth cone, it can add too much sensory data to the CNS. So in order to prevent that, we're going to need to do some sensory filtering, and the body will do this by using the lateral geniculate nucleus to increase the inhibitory filtering, and this will just prevent the hyperstimulation.
I had a question about the application part. You said it's going to be used to like for sterilization. How accurate is it compared to like the wavelength? Because UV has different wavelengths, so how accurate would it be compared to the UV light they use in hospitals and the UV vision?
It wouldn't match the accuracy, honestly, probably because there's multiple wavelengths that highlight different materials like proteins, bodily fluids, and bacteria. So the UV vision on a person would only see small parts in comparison to like the professional UV equipment.
Okay, I had a question. I wanted to ask, how would the system detect when UV levels reach threshold and then initiate retraction of the new lens?
So this would be done through like the UV cones. So overstimulation of the cones would go through the normal optic tract, and then all this overstimulation would lead to like an excess neurotransmitter release, and then that would send a signal back down to the oculomotor nerve, which controls the actual muscles, and this signal would be inhibitory, so pretty much stopping the constriction of the muscle and dropping the lens back down to protect the eye and block the UV light. Thank you, guys.